Autism spectrum disorder and Alzheimer's disease seem to occupy opposite ends of the human neurological lifespan. One is a neurodevelopmental condition manifesting in early childhood, while the other is a neurodegenerative disease characterized by late-stage cognitive decline. Yet, despite this massive chronological gap, these two conditions present with remarkably similar clinical deficits. Patients with both disorders frequently experience severe language impairment, motor problems, and extreme executive dysfunction. Look at their genetic architectures, and the divide widens again. Autism is linked to hundreds of distinct rare risk genes, while Alzheimer's pathology is driven by highly specific protein aggregations like mutant tau and amyloid beta plaques. But recent multi-omic studies, which analyze a biological system across multiple layers like DNA, RNA, and proteins simultaneously, reveal a hidden link. These vastly different genetic lesions actually converge on a single shared molecular bottleneck. Discovering a shared bottleneck means that a developmental disorder and a degenerative disease might be vulnerable to the exact same pharmacological interventions. That shared bottleneck is the hyperactivation of the Akt-mTOR signaling pathway. mTOR, or the mammalian target of rapamycin, is a highly conserved kinase that regulates cell growth, proliferation, and survival in the brain. To prove this convergence, researchers examined 3 distinct genetic mouse models. 2 models represent autism, driven by mutations in the Shank3 and Cntnap2 genes. The third represents Alzheimer's, driven by the mapped P301S mutation, which causes toxic tau buildup. Verifying that these 3 separate genetic starting points end up at the same destination requires synthesizing 2 entirely different sets of advanced biological data, SNOproteomics and RNA sequencing. By overlaying these data sets, we can map the precise route from distinct genetic lesions down to a shared hyperactive kinase cascade. If multiple distinct genetic faults lead to the exact same cellular failure state, then treating the failure state rather than the initial genetic fault becomes the most viable clinical strategy. We begin with a dataset from Mensur and colleagues, utilizing SNOTRAP-based mass spectrometry on the cortices of both the Shank3 autism model and the P301S Alzheimer's model. Mass spectrometry is an analytical technique used to measure the mass-to-charge ratio of ions to identify and quantify specific proteins in a sample. They looked at protein S-nitrosylation. Here, a nitric oxide molecule covalently binds to a cysteine thiol group, altering the protein. Comparing the SNO-proteome of both models, exactly 51 specific proteins were found to be S-nitrosylated in both diseases. Among these shared targets are crucial synaptic-associated proteins, such as GAP43 and CAMD2, which control axonal growth and synapse formation. The overlap also includes sensory receptors like P2RX3. Analyzing the biological pathways enriched by these 51 shared proteins reveals a stark conclusion. The data points directly to the mTORC1 upstream signaling network, achieving strict statistical significance. The shared pathology isn't happening at the DNA sequence level. It occurs at the hidden layer of post-translational modifications, altering the exact same regulatory network. Let's look at exactly how this hyperactivation occurs. In the autism model, S-nitrosylation specifically targets and modifies the TSC2 complex. Under normal conditions, TSC2 acts as the master biological brake on the mTORC1 pathway, keeping cell growth in check. When nitric oxide binds to TSC2, it impairs its pairing with TSC1. The brake is removed, leaving mTOR constitutively active. In the Alzheimer's model, S-nitrosylation targets the Wnt pathway, WNT11 and frizzled 10. This completely bypasses TSC2, achieving the exact same hyperactivation of mTOR. Mensur provides biochemical validation for this mechanism using Western blots, a laboratory method used to detect specific protein molecules. We see dense, thick bands of phosphorylated RPS6, a downstream marker of mTOR activity, in both mutant cohorts compared to faint bands in wild-type subjects. Whether the biological brake is cut in autism or the gas is pressed in Alzheimer's, the resulting cellular catastrophe of unchecked mTOR signaling is identical. Does this convergence hold up when we test a completely different genetic lesion for autism using a completely different assay? A separate study by Shank and colleagues tested this by running RNA sequencing on the hippocampus of mice missing the CNTNAP2 gene. RNA sequencing takes a snapshot of the entire transcriptome, showing exactly which genes are turned on or off at a given moment. This scatter plot details the gene expression data. Among the clusters, we see a significant upregulation of the MET gene and simultaneously the FNA5 gene. This transcriptomic shift triggers the PI3K Akt signaling cascade directly upstream of mTOR. To confirm this transcriptomic shift translates to the functional protein level, we look at the Western blots. The CNTNAP2 knockout column displays thick bands for phosphorylated Akt at the serine 473 site, compared to faint bands in the wild-type control. This cascade flows downward, confirming the final step, hyperphosphorylation of the mTOR complex itself at the serine 2448 site. Look at the total weight of the evidence. We have proteomics in the Shank3 autism model, transcriptomics in the CNTNAP2 autism model, and proteomics in the mapped P301S Alzheimer's model. Whether the disease is developmental or degenerative, and whether we measure RNA transcription or protein modification, the system fails at the exact same Akt-mTOR node. This brings us to a clear pharmacological hypothesis. If mTOR hyperactivation is the shared bottleneck, blocking it directly should yield a phenotypic rescue. Shank and colleagues tested this by administering rapamycin, a known mTOR inhibitor, alongside LY294002, an Akt inhibitor, to the Cntnap2 deficient mice. To measure the results, they used the 3-chamber test, an assay designed to evaluate rodent social cognition. The baseline behavior is stark. Untreated mutant mice fail to show a normal sociability preference for interacting with other mice over inanimate objects. This chart shows the sociability data post-treatment. Following the Akt and mTOR inhibition, the preference index of the mutant mice normalizes perfectly, rising to match the exact levels of the wild-type mice. Chemically modulating a single intracellular signaling hub can successfully restore complex whole organism social behaviors. However, we must be rigorous about the limitations of this data. This pharmacological intervention is highly specific. It is not a universal cure. This bar chart graphs the results from the open field and grooming tests. It reveals that rapamycin had zero effect on the mutants' severe hyperactivity or their repetitive behaviors. The distance traveled remains persistently high across both the treated and untreated mutant groups. Furthermore, there is a severe temporal limitation. The sociability rescue entirely vanished just 7 days post-injection. The mechanism of this failure is rooted in the genetics. The underlying architectural changes driven by the root genetic lesions remain completely intact. Once the drug metabolizes, the broken genetic code constantly pushes the system back towards dysfunction. Systems biology proves that diverse neuropathologies converge on targetable hubs like mTOR, offering profound behavioral rescues. But until we correct the root genetic architecture, these downstream interventions will only ever be temporary patches on a broken network.